So hi everyone, and welcome to our second Faba Bean Micro Summit. My name is Janelle Carlin. I'm the Director of Quality and Processing at Pulse Canada, and I'm going to be moderating today's session alongside Julie Mann, who is the founder of Appropriately Rogue Consulting. We are excited to have you join here today to review and discuss latest research on opportunities for Canadian Faba beans that has been conducted as part of the Pulse Science Cluster. So I just wanted to say that this research was made possible through a joint investment by the Canadian Pulse industry as well as Agriculture and Agri-Food Canada. So before we begin, I just wanted to address some housekeeping items. You can chat with other attendees or ask questions using the chat panel that's located on the right-hand side of the stream. And we've reserved time at the end of the webinar to answer questions, and you can address those to all of the speakers today. So just make sure to type in which speaker your question is for. We will be recording the webinar today and circulating it to all registrants after today's event. As well, in the area below this stage, you can find out more information about the Pulse Science Cluster, including speaker bios for today's event. And in this location, you'll also find a new report that highlights all you need to know on Faba beans. It's the first of a Pulse 101 series that is being developed by Pulse Canada. So first off, I'm going to be passing things off to Julie to make some opening remarks on the opportunities for Faba bean ingredients in the food sector. Julie is a passionate and connected food science and research development consultant focused on plant-based ingredients, finished products, and education. She was previously the Chief Innovation Officer at Purus Holdings, LLC, and her responsibilities include managing the R&D innovation process while developing and executing new technologies in finished food and beverage products. Julie was also responsible for identifying new business opportunities and innovative strategies in the growing market of plant-based foods. The integration of this role enabled new capabilities, new architecture with new partners, business models, and unconventional industry structures to educate and serve those opportunities, all while driving growth in the plant-based arena. So before this, Julie worked both with Ingredion as well as the Hershey Company, and she has a master's degree in food science and nutrition from Penn State and Drexel University. So with that, I'm going to pass things over to Julie for the opening remarks. Thank you.
Perfect. Thank you, Janelle. Nice to see everybody today. You're going to hear some great talks around the structural characteristics, bioactives, functionality, and the sensory around Faba. And so my point up front here is I'm going to be pretty brief, but I'd like to just share a few thoughts around Faba and the opportunities in the food industry. Many of you probably are very well versed in this space and understanding kind of what the market drivers are. So I'm not going to go into anything that specific, but just a few comments around plant-based food sales, how they're growing. They continue to outpace the total food market by about 3 times. And so that's something really important because Faba is going to fit. It's going to play a role within that plant-based food sales and within that kind of growing and burgeoning market. So if we think about all the categories right now that plant-based is playing in, you can barely name a category that plant-based is not playing a significant role. And really, I think Faba's opportunity is to start leveling up. And as more crop and more product is available, bringing the increase of Faba bean as a percentage of market will be essential going forward. And I'm not going to read these slides to you, but it's more to say, the opportunity is there. We just have to kind of grasp it. And also the Faba trend is global. It's not a Canadian or a North American play. We're seeing the rise of ingredients going into finished products and the acceptance. That's the key. The acceptance of consumers of these products. Really, mostly in Europe and North America, it's been a little bit flat in some of the other areas based on a lot of kind of socioeconomic reasons. But we're seeing the same trends. This data is only up to 2021, but we're seeing the same trends coming forward for 2022. So you can trust that Faba is it's on the global movement, which is a good thing. So some of the top categories where we've seen the growth is really around that hand-to-mouth vegetable snack. So that whole bean going into a finished product, so sort of like a Three Farmers product. Also bread and bread products, and some meat substitutes. I would say the majority has been based around whole bean or flours, because the concentrate to isolate market is building and it's growing and it's doing great, but it's still a little bit early on. So there hasn't been, the excessive supply of fava like there is pea or soy or some of the other proteins. So I think we'll continue to see this lens change a little bit. We'll see new things being added, tofu, other bakery snacks, other dairy replacement areas. And all ingredient formats are playing. So flours, whole bean, concentrate, isolate, they're all playing in the market. And it's important for us to understand that, because really those are the bread and butter of what we're working with, right? To help the industry innovate. And we have to make sure we understand, again, as you're going to hear today, a lot of the clarity around the different buckets, whether it's a whole bean, a flour, a concentrate, an isolate. And here you can just see, some of the traction is really in the labeling around fava bean and fava flour, a little bit around concentrate and paste. But when it gets to the isolate, again, I think it's something that we're still in the progress of building, right? So I only have one or two slides here that I want to share left, and really it's that we're working on an industry that the ingredient opportunities are endless. From the whole bean to the isolate, yes, we know that the whole bean is just that. The flour is a dehulling and milling operation. The concentrates are an air classification. The isolates go more into, a wet isolation, fractionation process. Each of those has very specific functionality, sensory attributes, nutrition that goes very specifically into individual segments. And that's where the unlock is for Faba. We have to just really, as an industry in this space, and all the partners who are working in this space, get really good at understanding which ones work for the best segments, or how do we best leverage. So my last slide is, there's really been, and I'll leave you with this, and then we're going to get into some great research that's being done today. There's a fundamental shift going on in the plant-based industry. When I started many, many years ago now, we were just leveraging plant-based ingredients because the movement was there and we wanted to get plant-based into foods, right? So we introduced a lot of proteins, formats, types, sources. We started to build some consumer awareness. Where we are now is that consumer has gained their priority. They're telling us what they want, what they need, and we have to deliver. So if you look sort of from the left to the right of the slide, once consumers' needs were raised, we realized that, we have to do better. We have to make it better. We think we're doing a great job, and in a lot of cases we are, but we always have to keep pushing the envelope just a little bit further on organoleptics, on functionality, on clean and simple, sustainability. And that's going to require this characterization, research and development, innovation, and process development to really help us to deliver what we need for the consumer. So today, again, you're going to hear some great research from a lot of folks, and we're going to end my portion now, and I'm going to get ready to introduce our first speaker. And our first speaker is Janatha Wanasundara. She's a senior research scientist with Agri-Food Canada. Her research program is in protein chemistry and byproducts, and she focuses on oilseeds and pulses. And she has published over 90 peer-reviewed journals. I was astounded by that. Congratulations, by the way. And books and chapters on bioactive ingredients from plant sources. She's also an adjunct professor at the University of Saskatchewan and contributes to a lot of graduate student training, and the co-editor of Sustainable Protein Sources, which is a book that is going through its second edition. So welcome, and we're excited to hear about what you have to tell us.
Thank you very much, Julie, for the kind introduction. My presentation is on understanding faba bean seed structural characteristics and dry fractionation. It is what I will be presenting today is the first object, what we learned from the first objective of this Pulse Science Cluster project you mentioned, that the title is Strategic Approaches for Faba Bean Value Addition and Utilization. As Julie spoke, there are good things, many good things about faba bean, and then this is how the project was developed. And my co-investigators of this project, Drs. Lazin and I and Shant, will be presenting about other three objectives of this project. As you mentioned, as we heard, faba bean had very good, many good things. And first advantage that producers have is as a pulse, it has high yield potential. And at the same time, faba bean is high in protein among the pulses available today around us today. Therefore, per unit area, plant protein production or pulse protein production is comparatively high. So we can use faba bean as whole seeds, as mentioned. That is what the majority of use. But to get faba bean into food application, we need to develop ingredients. That is what Julie also mentioned. And in here, what I'm showing here is we need to get whole seeds, the raw material, fabricated into certain level of ingredients, and then go through modification to suit it to the perfect application that it goes. In the first objective of this project, what we looked at is dry fractionation as mode of fabrication for faba bean available in Canada or grown in Canada. To touch base on dry fractionation, it was mentioned before. Dry fractionation take the advantage of a natural architecture of the seed, the cotyledon. The picture here shows the ultrastructure of pulse cotyledon, and starch and protein are the main components that we can find that are stored in these cells, and those are the valuable components of faba bean, similar to other pulses. So starch is in its discrete granular form in the granule, and proteins are deposited around the cell. And what happens during dry fractionation is seed coat is removed and seed is milled. Seed is milled means broken into pieces. So the fragmentation lines during milling can take the natural contact lines along the starch granules. So that way, large pieces and small pieces are become available from milling, and depending on what is the composition, their size as well as the mass is different. So when they are subjected to an airflow, they have different velocities, so that way the large particles or heavier particles that are rich in starch is separated from lighter small particles that are rich in protein. So that is the advantage this process has. And this Sankey diagram published by Schutz and group from the Netherlands show clearly clearly the use of energy and mass in other form of protein product preparation of sorry, uh, available, that is wet fractionation. So clearly, compared to wet fractionation, dry fractionation does not use water and it has low energy requirement. At the same time, because of no other additives or uh, solutions or anything is used, the material, protein and starch in the final material that is obtained for dry fractionation hasn't gone through much alteration, so they are in their native form. The disadvantage is compared with fractionation, protein or starch coming from dry fractionation is less refined, so we can't expect much purity. The study involved using 3 different varieties grown in Canada. The reason to select these 3 different varieties is the seed size and their chemical composition. Malik, which is in the very right hand corner. They are the large seed size faba bean, a traditional variety grown, and it has seed coat tannin level at regular level, so as the lysine and conlysine at regular level. Lysine and conlysine is special to faba bean. It is found in faba bean and it has an antinutritive property. The one in the middle is snowbird, which is lighter in color. It's almost white, grayish white, and because of low seed coat tannin, and it has a small seed size, almost close to a yellow pea seed. And lysine and conlysine level is at regular level. The one in the very left-hand corner is Fabel. Fabel is bred to have lowest lysine and conlysine level available in faba bean grown in Canada. And it has medium-sized seed and it has regular seed coat tannin level. So there are 3 different types of seeds that had been grown in 3 different locations in Saskatchewan in 2 years and went through dry fractionation at Canadian Grain Commission. And the conditions that is available that was used for dry fractionation is optimized for faba bean. And fine fraction and coarse fraction was obtained from this dry fractionation of each seed sample, and yield data, composition data were obtained. And this one graph shows the protein content. So this whole data I will be presenting is on the fine fraction, which is the protein fraction. But how much protein was available in these seeds to be fractionated? The spread is in the graph, and it showed 29% to 35.5% protein was available in D-hull faba bean flour from these seeds. And the lowest value and the highest value came from 2018 samples grown in 2 different locations and from 2 different varieties. What does that mean? So we can expect, if all protein is transferred into the fine fraction, and fine fraction it contains 100% protein is transferred there, we can get 29 to 35% of flour dry weight as the fine fraction. This slide shows three graphs, that is on the dry matter yield and protein content and starch content of fine fraction and coarse fraction. The one in the far left-hand corner, yellow and orange color, is for fine fraction, and the green color fonts is for and symbols are for coarse fraction. And Y1 is 2018, Y2 is 2019. Yeah, and it is clear the yield that that was able to obtain for all these seeds were between 24% to 29% between 2 years, and fine fraction yield was around 69 to 75%. And the protein content of fine fraction is between 64% to 71%. And there was some starch contaminated in that fraction. It was at 1.5 to 2.7%. Some of the protein in whole dehulled flour was fractionated into coarse fraction, and it was about in the coarse fraction it was about 17% to 23%. And using these numbers, we were able to calculate some of the meaningful numbers. In the graph, in the table, the black color fonts indicate the dry matter yield, and blue color is protein yield. In 2018, this is how we did that. Faba, each variety grown in all three locations were pooled together. And if we take 100 kilograms and going through dry fractionation, what it would be like, what numbers we can obtain. 2018, doesn't matter what the variety is, we can get definitely 25 kilograms of fine fraction. But each fraction, how much protein is transferred, it was around 17% protein. 17 kilograms of protein we can obtain from these dry fractions. And 2019, the numbers varied. Using that, the efficiency of the protein fractionation can be calculated, and except Malik and all others were between 53 and 55% protein separation efficiency. These numbers were, when we checked what was reported for PP, dry fractionation is the available literature for comparison. It comes as 49 to 51% protein in the flour can be shifted to fine fraction in P. So Faba bean is comparable or much better than P. And lysine and conlysine level is has a negative aspect, brings negative aspect to Faba bean. Because we had very low lysine conlysine containing Faba and wanted to see whether it is going into the fine fraction. And the blue table shows what the levels in the dehulled flour, and green table shows the spread of range of data that is present in different locations. And Fabel spoke to its quality trait, and there was low amount of lysine and conlysine, but it was transferred into fine fraction. And Snowbird had the highest lysine conlysine level. So from that, what we understood is lysine and conlysine has been in the lighter particles of the seeds. That means there may be association with the protein or where the proteins are deposited. The other important one is oligosaccharide levels. Although we found raffinose, stachyose, and verbascose, all three oligosaccharides in dehulled flour, only in the fine fraction we found stachyose and verbascose only. And it is clear that verbascose did not show a pattern, but stachyose showed a good pattern between years and the location. The other negative antinutrient everybody talks about is phytate levels, and phytate levels spread, all these samples were spread between 7.5 to 28.5 milligrams per dry matter. And in the P, the reported values by Ning Wang's group from Canadian Grain Commission for dry fractionated yellow P is 10 to 15 milligrams per gram. These are for the initial functional characterization of Faba bean, fine fraction obtained from these seed samples. And what is the more key is water holding capacity of Faba bean fine fraction was between 0.65 grams of water per gram dry matter, which is comparatively a low value compared to other protein concentrates obtained from P under same dry fractionation conditions. Oil absorption capacity was also between 0.8 to 0.9 grams of oil per dry matter flour, which is also at the lower end, quite a little bit lower compared to P. Oil emulsification capacity was a little bit higher, although it showed a considerable variation between years, varieties, and location. And about between 340 to 380 milliliters of oil can be emulsified per gram dry matter. This slide shows statistical significance of data and what we can learn from it. Flour protein content, that is the important part. How much protein is in the seed, whether these factors affected the protein content. Yes, year, location, and variety affect the flour protein content. And then going through dry fractionation, whether variety is a considerable factor. No, it's for the yield and the fractionation efficiency. It's the year and location, mostly the environmental factors that affected the fractionation. And in the fine fractions, the protein content definitely affected by year, location, and variety, because starting seed material or the raw material protein content is important. But starch content in fine fraction had no bearing on these from these factors. And for antinutrients, we present here phytate, year and location. There's no variety effect. And for lysine and conlysine, because we worked with the low lysine conlysine, and so the variety has an effect, and also the environmental factors. So as for the stachyose level, and verbascose was not affected. For the functional properties, water holding capacity and oil absorption capacity, both are affected by year, location, and variety. And oil emulsification, only location and the variety. As conclusion, we worked on this value in this value chain. Raw material conversion, fabrication using dry fractionation. So the process, considering the process, protein partitioning into fine fraction is more related to the environmental location and year factors. That means either small seed, snowbird, or large seed, Faba, that goes through dry fractionation, it can give the similar yield or the fractionation efficiency. But the growing environmental conditions are the one that affect. In the protein-rich fraction, high protein content was in 2018 and then 2019. And also considering the varieties, Faba and Malik consistently give high protein level compared to snowbird. Although they are 2-3% differences in numbers, when it comes to large-scale fractionation, that matters in the ingredient quality. And so for more protein in the flour, more protein in the fine fraction. In terms of antinutrients, lysine, conlysine, phytates, and oligosaccharide are also transferred to this fraction besides protein and starch, and particularly lysine and conlysine. And low water holding capacity and oil absorption capacity of the fine fraction can be taken to advantage to utilize or modify further for use in further applications. I would like to acknowledge funding for the project through Pulse Cluster, which is from a partnership with Canadian Pulse Industry and Agriculture and Agri-Food Canada, and technical support came from Agriculture and Agri-Food Canada, Dan Sutherland and Debbie Anderson, and also co-op student Joshua, and research teams of Canadian Grain Commission, Dr. Ning Wangs, and Benjamin Tran, University of Saskatchewan, who grew the Faba bean plant for us, was very much acknowledged. And initial discussion with the industry, contribution, wealth of knowledge provided by Dr. Mehmet Tolbek and Chris Chevilla also acknowledged. Thank you.
Right, thank you so much. I'm now going to introduce our next speaker here. So we have Dr. Lamia Lucien. She's a research scientist at the St. Hyacinthe Research and Development Center of Agriculture and Agri-Food Canada in Quebec as of 2008. And her research activities focus on plant proteins from oilseeds, legumes, and cereals, their extraction, characterization, as well as the evaluation of the impact of processing on the molecular, nutritional, functional, and allergenic properties of using in vitro and cellular models. Her research aims to promote alternative and sustainable sources of proteins by demonstrating their health benefits and by developing new added value and functional protein ingredients. Her education includes a Bachelor's of Food Science from the National Institute of Agronomy, a Master's and a PhD in Food Science and Technology from Jiangnan University, and she also completed 5 years of postdoctoral training, 2 at McGill University and 3 years as an NSERC postdoctoral fellow at Agriculture and Agri-Food Canada. So with that, I will pass it along. Thank you.
Thank you. Thank you for the kind introduction, and also thank you for the opportunity to present some of our research results on the nutritional and the bioactive properties of the Canadian faba bean protein. So I won't go too much on detail in introducing the importance of faba bean. I think my predecessor's speaker has done a very good work on that. But I would like just to maybe emphasize that faba bean has also, in addition to its high protein content and agronomic advantages, some also health promoting properties through many bioactive components, including phenolics, L-DOPA, GABA fibers, and peptides, which has actually less studied for faba bean and which is one of the objectives of our research. So in this project, we have our objective was really to get more information on the nutritional and health promoting properties of Canadian faba bean varieties, which are less studied among other varieties, for example, coming from Europe. So we choose 3 varieties, which are the Malik, the Snowbird, and the Fable. Some particularity about these varieties, for example, is their difference in the tannin content, or the Fable, which has a low vicin and convicin content, which is of course of interest for utilization for people who have, who suffer from favism. So it's very important, of course, for the utilization of any new source of alternative protein, or even to use protein-derived ingredients, to know their protein quality. So the protein quality is defined, of course, not only by the amino acid profile, and also the amount of the essential amino acid that are brought by the faba bean, but also it will be influenced by the internutritional content, compound content, and also the digestibility. But in addition to considering only nutritional quality, it's more and more considered in the protein quality the health beneficial bioactivities that are brought by this protein. And this is generally represented by the bioactive peptide that are released after the digestion of these proteins. So the methodology that we have taken in this work is to transform the faba bean seed into flour, and to process them as to simulate what will happen after processing. And we choose the very common process, which is boiling in this situation. We compared faba bean to very conventional and most used other source of protein, which has pea and soy. And we have these flours, after treatment, have undergone in vitro gastrointestinal digestion. In this situation, we used the standardized and harmonized InfoGest protocol, which is a multi-step gastrointestinal digestion. We simulate the digestion both in the stomach, in the like the stomach and duodenum. And because we aim to assess the nutritional quality using to determine the DIAS, which is the digestible and digestible amino acid score, which has been the protein quality method that has been proposed by the FAO to replace the PDCAS, because the DIAS will look into the individual amino acid as individual nutrient. So it's very important to determine the digestibility of each amino acid into the ileal phase, which is a much better estimation of the protein quality than the PDCAS that look at the protein digestibility as a whole over the whole digestion tract. So to achieve that, we did modify the InfoGest protocol by adding a jejunal ileal digestion phase. So after the digestion, we collected the digestates of the different samples and undergone amino acid analysis to determine the different scores. So as I mentioned earlier, protein quality is also affected by the antinutrient content. So we determined, one first step was we wanted to determine the different antinutrients, particularly the phenolics, which are not considered necessarily antinutrient, but they do have an impact on the digestibility of proteins. So this will have definitely some impact on the final protein quality. Of course, the trypsin inhibitor activity and the phytic acids, which also bind to proteins and reduces digestibility. So as we could see from this table, there was no major differences in the protein antinutrient content among the faba bean variety, but we can see that, for example, soybean has a much higher content of trypsin inhibitor activity than faba bean and pea, and also we can see that phytic acid is much higher also in soy compared to faba bean. Pea in this situation had the lowest phytic content. In terms of protein profile, there was also no differences between the three varieties. They had essentially the same protein profile, but we can see from this figure on the right that the boiling had an impact on the protein profile. So, and we can see after boiling, the formation of some protein aggregates, so that we think might also have an impact on the digestibility of protein. So in this slide is presented the amino acid score, which is actually a measure on the capacity of the protein to meet the needs in essential amino acids. So it's a measure of the milligram of essential amino acid in 1 gram of protein versus the need for a reference protein. And in this situation, we have used the needs for children from age of 6 months to 3 years, which is recommended by the FAO. And we can see from this figure that faba bean protein actually meets the needs for most of the essential amino acids, but of course, which is also unknown for the sulfuric amino acids, methionine and cysteine, which has a little bit in lower content in faba bean as most of other legume proteins. We can see also that soy has also is superior in terms of protein in terms of protein profile by bringing a higher amount of essential amino acid compared to faba bean or pea. But this is very known that soy has a very equilibrated amino acid profile. We can see also some differences between the amino acid score after treatment, and we can see, for example, that after boiling, the the valine content is lower than what is required. So here we can see the digestibility of the whole amino acids, and we can see that there was no differences in digestibility among the 3 faba bean varieties, and they were also comparable to pea and soy. And we can see here that soy has seems to have from our data a little bit lower digestibility, which could be attributed to the higher content in trypsin inhibitor. But also we can see reduction of digestibility after boiling. Then again, this will depend on the condition used. For our particular condition, we observed a reduction in the digestibility, which could be related to the formation of the aggregate, and then there is the enzymes have less access to the protein for the hydrolysis. So after assessing in the digestibility for each amino acid, we can see which is the most limiting amino acid, and it's based on the most limiting amino acid that which is represents the dias. And in this situation, we could see that there was no big differences between the 3 faba bean varieties, but the Fabel seems to have a higher in vitro dias than the other 2. And also the dias were very similar to what is obtained for soy or pea. Of course, the dias we have compared them to other protein which has known to have a very good protein quality. In this case, it's whey protein isolate and casein from milk, and we can see that, of course, the dias for faba bean and for other pea or soybean are lower than those for casein, but which is also very known that animal protein has also, they have a very good protein quality and very high dias compared to plant protein. But what we can see from our data, of course, we use in vitro study in this to generate these dias, and we compare them with what is available in vivo, because of the difficulty to carry on animal study. We wanted to see if the fact that using in vitro digestion model gives comparable results. And we can see, of course, they are much lower than what is obtained in vivo, but there was a very good correlation between the in vitro dias data that we obtain and the in vivo dias that are available in the literature. So we have a positive correlation of over 0.87, which is very, very good. So that means that the in vitro digestion system is very valuable tool to assess protein quality of faba bean, but also for all other plant proteins in comparison to using in vivo, like in vivo studies using animal, due to, of course, cost, but also some of these ethical concerns. So in summary, the Canadian faba bean have a very high protein quality, which is very much comparable to other pulses and all seeds, and also, of course, that the used InfoGest digestion protocol can lead to determine diases value that are very comparable to in vivo model, and it's very useful tool when in vivo models are difficult to get or not available. So we pass now to the second component of our presentation, which is talking about the bioactive properties of faba bean, looking particularly to most important like health promoting bioactivities. So of course we use the same samples, and also they undergone the same gastrointestinal digestion, and we after the digestion we enrich the digestate to get the most bioactive peptides, and determine their different bioactivity by using in silico, in vitro, and both cellular model to get closer to physiological conditions. And we particularly look to antihypertensive, anti-diabetic, and antioxidant properties, which has very important, particularly that these non-communicable diseases are really prevalent in the population. So for the in silico analysis, the in silico is that we is kind of theoretical assessment of the bioactive potential of the protein, and there are some tools that are available online that allow to do that, and they will depend on how much bioactive peptides have been identified or characterized by the different research groups, and that they enter their data into the system. So what appears is that by simulating what is the potential, the bioactive potential of the different Faba bean proteins, it appeared that most of the protein have either anti-diabetic or also anti-hypertensive inhibition capacity, but also they have also antioxidant capacity, although it appears not very low, but most probably because it has not been studied that much as the other two activities. But it's a good indication on where we have to put efforts to study the bioactivities. By using in vitro assays, we determine the antioxidant activity, and in that we use different assays that target different type of mechanisms, either electron transfer or hydrogen transfer antioxidant, but also some of the iron chelating mechanism that will chelate the formation of free radicals. So what we can see here is that Faba bean, they have a very high DPPH activity, but all in comparison, for example, to pea, but they are very quite similar to that of soy, while they have also a high ABTS activity and ORAC and iron chelating. Of course, soy here, these data are IC50, so the lowest the value, that means the higher the activity. So in this situation, for example, soy has a higher iron chelating activity, but he has also a higher ORAC activity, because here is in concentration. So I hope this is not too much confusing for you, but the main conclusion we need to retain is that Faba bean has a very good antioxidant activity, which in this situation seems better than that of the pea that was used for this study. Also, when we compare with we use the cellular antioxidant assay, which is a cell using the CACU2 cell, which is most closer to the physiological effect in humans. So it appears that Fabel has a very interesting antioxidant activity in comparison, for example, to Malik, and similar also to snowbird, and much higher to pea, and also higher than soy. So this is also a very interesting result. Regarding the anti-diabetic and anti-hypertensive activity, we could see also that the Faba bean has a good anti-diabetic activity, but similar to the other. The soy remains to have the highest anti-diabetic activity compared to Faba bean. But for the ACE inhibition, also the Faba seems to have an interesting antihypertensive activity, and particularly the Fabel1. So because Fabel stood for its high antioxidant and antihypertensive activity, we decided to continue the work with further with this variety to fractionate further the different peptides that appeared, and to enrich the different fraction to identify those peptides. It was important to us because, of course, the digestate, in this situation, we digested flour. So the flour may contain other components like polyphenols or polysaccharides that could play a role in the bioactivity that we could see. So by doing this work, we wanted to ensure that the bioactivity that we see is related to the peptide and not to the other components. So we did the enrichment of these different fractions, and we analyzed those fractions, and we could see, for example, that the different bioactivities, I'm looking for the word in French, but we can find them in the different fractions. For example, so the F1 actually was richer in the ABTS fraction, while the F3 fraction that contained another type of peptide was richer in the iron chelatin bioactivity. But the F1 contains also most of the antihypertensive activity. So after analysis by mass spectrometry of the different fractions, we could identify at least 10 peptides that are known to have ACE inhibitor or antioxidant activity by using by using the different in silico data. So this is confirming that the peptides that have been identified by MS are bioactive peptides with the bioactivity that we have been assessing. So these peptides, we have sensitized them actually, and to get larger amount of these peptides, and also to confirm that these peptides have these bioactivities. So after sensitizing this peptide, we carried again the various bioactivity tests, and it appeared that, for example, some peptides had higher DPPH activity. Most of these peptides actually had high DPPH activity, or sometimes the same peptides that have antioxidant activity, they also had ABTS activity. For example, these two peptides had a very high also ORAC activity. So they are very, very good antioxidant peptides. Similarly, those peptides, for example, these peptides have between 8 to 10 amino acid in length, which is very accurate to what is published in the literature. So these also had a very good antihypertensive activity. So it confirmed that the bioactivity that were observed from Faba are really due to the peptides, which increases the quality of the protein. So just to summarize what I have said is that the bioactivity assay, of course, revealed that Faba flour, when digested, had a very good bioactive properties, which appear to be better than those of pea, and slightly lower than soybean. But the conclusion is that Faba has a very excellent health promoting potential, in particularly antioxidant and antihypertensive, which complements very well its nutritional quality, and therefore present a high potential for use in the development of new functional or nutraceutical ingredients in food applications. So that concludes my talk. But before ending, I would like to acknowledge, of course, my team. First, Delphine Martineau-Coté, who is the PhD student who has been doing all the experiment work, and this work is part of her PhD thesis. And she is affiliated to McGill University, and our professional Dr. Alawa Ashuri, my two research assistants, Melanie Pitre and Stéphane Sirois, and of course collaborators, particularly Janita Wanasundara, and finally the financial support of the AgriScience Post Cluster for funding this work. So thank you for your listening, and I will be happy to answer any of your questions. Thank you.
Great. Thank you so much. We're gonna move right on to the next speaker. Our next speaker is Yang-Feng Ai, and he is an associate professor and Saskatchewan Ministry of Agriculture endowed research chair in the carbohydrate quality and utilization in the Department of Food and Bioproduct Sciences. And that's, of course, at the University of Saskatchewan. The primary goal of the work that Yang-Feng is doing is around carbohydrate chemistry and utilization, really to promote value-added products with carbohydrates in food, feed, and other bioproducts. And I think, from what I've gathered, his research really includes carbohydrate chemistry, but it also includes nutrition, value-added processing of cereals and pulses, and other biomaterials. So Yang-Feng, we're excited to have you, and take it away.
Thank you, Julie, for the nice introduction. Today it is great for me to share some of our recent research on faba bean starch. And as you already heard from previous speakers, starch is actually the leading component in faba bean. This is very important post-crop in Canada. For my presentation today, I will start with an overview of the faba bean in the post industry. Actually, a lot of information on my slide already covered by previous speakers, so this can save us some time. I will focus on some of the things not covered. After that, I will move to talk about the granular and molecular structure of faba bean starches from 5 different varieties. And then in the end, I will talk about the functional attributes and also in vitro digestibility of faba bean starches in comparison with the commercial pea and also maize starches. The main use of faba bean in North America, when we look at the crop in the continent, actually the main use still for feed, may be surprising to you. So we can see it can be used for all the different animals. When we look at the food application, I know a lot have been covered, but I think the whole seed as a canned product is still, it's a conventional product, but I think it's still one of the main use of faba bean in the market. And in the food store, you can see them maybe called as a broad beans or as a faba beans. Those are we are talking about the same crop, but different names. Also, I think Julie already mentioned it can be milled into flour. I'm not going to talk too much about that, but you can find like the product on the right from the Bob's Red Mill. They produce they call it faba bean flour. Definitely, we can use either dry or wet method to fractionate the like starch and protein, and which already nicely presented. I'm not going to repeat. This also covered by our previous presenter. The main reason faba bean has become like really popular, particularly in terms of the value-added processing process, is because of the high protein content. And because of that, proportionally, we see the starch content of faba bean lower than the pea in usually used for the different value-added processing. And both crop have about 15 to 20% of the non-starch polysaccharide. So these include like cellulose, hemicellulose, and some other functional and also nutritional polysaccharide like a pectin. But there are some challenges of using faba bean in the feed and also food industries. Some of these already covered, like the anti-nutritional factor. what I want to emphasize here is some of these anti-nutritional factors, they are common in different pulses, not unique to faba bean, like the inhibitors of the digestive enzymes, tannins, lectins, and also fatty acid. But the lysine and the convicin already nicely covered by Dr. Janita Valsandra. This is unique to faba bean. And then the health problem it can cause is called favism, because for human subject, like deficient in the glucose 6-phosphate dehydrogenase, they will develop some this symptom. Usually it has a symptom of we call it hemolytic, the anemia. So that's usually the symptom we're talking about, and sometimes can be very severe to human health. Another thing is the large seed size of the certain varieties of faba bean actually also make it difficult for the like feed and food application, because the larger seed size may require the equipment modification for seeding and also processing. And a very interesting I learned from farmers in Saskatchewan is because of larger seed size, and usually will lead to the higher seed cost. Because when the farmers they go to buy the seed, they don't go by the numbers of the seed they are going to plant this year. It's more by the weight. So I think this is also, if you look at the small and the large seed on the slide, the for one seed the weight will really vary quite a bit. So I think so when I first heard this from the farmers, it's kind of surprising to me, but that's the fact we are facing. I think this slide already nicely covered by Dr. Janita Wanasanto and also Dr. Lamia Losin, so I'm not going to repeat it. So it's really a collaborative project. We try to look at all the different components important for varialization of a faba bean. So for my talk today, I will mainly focus on one paper we published together last year to look at the structure, functionality, and also digestibility of faba bean starch. So for this study, we include five varieties of different faba bean, very popular, like cultivated or also processed in North America. Some of these already covered in previous talks, but we also have like a snowbird. This one very similar to snowdrop. They have a small to medium seed size. Also, we have tabor and the Faba and the medic already mentioned in previous talks. So I'm not going to really repeat, but you can see in addition to the differences in seed size, they also have different levels of the tannin and lysine and the convicin levels. After we obtained the seed from Dr. Wanasandra, we try to use a wet method in our lab to isolate the starch from the grains. And you can see if we look at the granular morphology of the starches under scanning electron microscope SEM, the faba bean starch and the pea starch, they belong to the same group. So the starch granules, they have either kidney shape or oval shape, spherical shape, and some of them have an irregular shape. But when we look at the granular morphology of the like a normal maize starch, it's very different. First of all, the granular, the shape, it's different from the pulses, because the maize starch, they have a polygonal, spherical, or irregular shape. And also the surface of the maize starch seems to be less smooth in comparison to the pulse starches. And also we can easily tell that the granular size of the normal maize starch is smaller than the pulse starches. We look at the X-ray diffraction pattern of the different starches. This is the like a technology we usually used in starch chemistry. All the pulse starches, they show a C-type X-ray diffraction pattern, which is a combination of an A and a B type polymorph. But for the normal maize starch, it only shows the A type polymorph. When we look at the crystallinity from the X-ray diffraction pattern, then the different starches, they show a percentage crystallinity from about 28% to 31.6%. And the lowest we find is in the snowdrop from 2016. For the normal maize starch, it shows a higher percentage percentage crystallinity than all the other pulse starches. When we look at the particle size, the different starches have a particle size range from about, on average, about 16 micrometer to about 30 micrometer. And then the normal maize starch show the smaller particle size, the average particle size than all the pulse starches. We determine the amylose content of the different starches. So for the faba bean starch and also pea starch, you can see actually the amylose content determined using iodine calorimetry, they fit into a range of 35 to 40%. This is a very typical range of amylose we find in the starch from the different pulse crop we grow in North America. In comparison, normal maize starch only have about 30% amylose. We also look at the gelatinization temperature, the gelatinization property of the different starches. The only thing I want to emphasize here is when we look at the faba bean and the pea starches, their gelatinization temperature are significantly lower than the normal maize starch. Particularly if you look at the onset gelatinization temperature, the faba bean and the pea starches, the onset gelatinization temperature is about 7 to 10 degrees Celsius lower than the normal maize starch. So why this is important? Actually, the paper industry has already taken advantage of the low gelatinization temperature of pea starch. They can use the pea starch as adhesive for the corrugated cardboard. So the low gelatinization temperature means we only need to cook the starch to a relatively low temperature before it can function as an adhesive during the corrugating process. So because we only needed to heat the starch to a lower lower temperature, so which means we can save energy, and also which means during the like gluing process, the machine can operate at a higher speed. So I heard from the paper industry, actually they already start using some of the pea starch for that purpose. But I believe the faba bean starch can have the same advantage in the sector. We also look at the retrogradation of the different starches after we store the sample at 4 degrees Celsius for 7 days. What we can see here is the faba bean and the pea starches, they show similar gelatinization percentage, significantly higher than the normal maize starch. So usually when we use the starch, like the faba bean and also pea starch for the different food application, the high retrogradation rate usually is considered as a undesirable attribute. As you can see on the slide, so if we see a higher retrogradation rate, so when we use the starch as a thickener, as a stabilizer, as a binder in different food, for example, like in yogurt, if usually if we have a higher retrogradation rate, which means we will see the phenomenon called syneresis. So we will see the expulsion of the water from this gel structure, which is usually considered as undesirable. We also look at when we heat the starch in water from 50 degrees Celsius to 95 degrees Celsius, how the viscosity will change as a cooking and also in the cooling process. So what we can see on this slide is when we compare the different pulse starches with the maize starch, you can see they have a lower pasting temperature, the lower peak viscosity, but most of them, they have a higher final viscosity. So this again confirms what I shared with you. The pulse starches, they require a lower cooking temperature in order to develop the viscosity. And most of the samples actually, between the 2 years, they show very good like consistency in their pasting profile. But there are two varieties, like a snowdrop and also snowbird. If we look at the 2 years, actually we see noticeable differences in their pasting profile. When we look at the amylose content, their branch and length distribution of amylopectin, their granular structure, actually until we publish the paper, we don't have very good answer to explain why they show such a big difference in their pasting profile. But for other 3 varieties, they didn't show this big variation in their pasting profile. So we also determine the gel strength after we cook the starch. So basically, after we cook the starch, we put it into a container and then store it at room temperature for 2 hours. Then we use a texture analyzer to determine the hardness or the strength of the gel. What we can see here is when you look at the gel strength, it is in order of faba bean starch greater than P, greater than normal maize. So the strong gelling ability of the pulse starch is well known in the industry. So in the past few years, we see the more and more the pea starch and other pulse starch are used for the different product, as you can see on the slide, like the glass noodles and also this jello type of dessert, and also gummy bear candies, and also it can be used as like the raw material for the soft capsule. All these are related to the strong gelling ability of the starches. And so we are very curious because even though faba bean and pea, both of them are pulses, but in the first study, we show that the faba bean starch, at least for the 5 varieties, show the higher gelling ability than the commercial pea. So is it really more like a fact for all the different faba bean and the pea? So in a follow-up study, we actually look at the isolated starch from different the faba bean varieties, and also we include 3 varieties of the pea. Usually we cultivate it in North America, like CD Striker, CDC Inca, and also CDC Meadow. We still include a commercial pea starch from the company. So when we look at the gel strengths, actually all the 3 faba bean starches, they consistently show a higher gelling ability than the pea starch. So we are still carrying out some other like the experiments to really look at the granular and also molecular structure of the different starches, and also to see how they change during the cooking. So it's really great to see the faba bean. The starch seems to have a better gelling ability than the pea. So which means for some of the mentioned application, faba bean starch may be more desirable. So in the last part of the study, we look at the in vitro digestibility of the different starches. This slide shows the starch without cooking. So what we reported here is a resistant starch. Resistant starch is defined as a fraction of the starch that can resist the digestion in our small intestine, then can be passed into our large intestine. So resistant starch is basically a new type of dietary fiber. When we look at the resistant starch content of the raw starches, we clearly see an order of the faba bean starch show the greatest enzymatic resistance, followed by the pea and followed by the normal maize. And in our paper, we nicely show that actually this is related to the branching length distribution of amylopectin and also the internal granular structure of the different starches. But after we cook the starch in a boiling water for 10 minutes, so we can see actually the resistant starch content are largely comparable. And the range is about 4 to 7%. So which means after cooking, the starch, they show similar digestibility. So here are some of the key takeaway message I want to share with everyone in today's webinar. One is when we compare the normal maize starch, faba bean, and pea starch, they show more similarity as they had high amylose content and lower gelatinization temperature, higher retrogradation rate. And also when we look at the pasting profile, the pulse starches, they have a lower pasting temperature, lower peak and breakdown viscosities, and generally greater final viscosity. So which means when we find one way to utilize the pea starch, the most of the time the knowledge and technologies can be transferred for the varialization of a faba bean starch to develop different ingredient and also product. But when we look at the gelling ability, we clearly see a trend of faba bean greater than pea and then the normal maize starch. So in certain applications, faba bean starch may be even better than pea starch. The resistant starch content of the raw starch also show a trend of faba bean greater than pea and then greater than normal maize. But this difference disappear after cooking. So last thing I want to emphasize here is for this study, we have the faba bean from 2 growing year. It seems like they didn't really change the structure and also the functional property of the starches. So overall, based on my experience, seems like the growing year, unless there's really a big change in the climate between years, but most of the time, different year we still see a relatively good consistency in the functional property of most of the starch. But for our study, we did see some like variation in the pasting profile of the snowbird and snowdrop faba bean starch, as already explained. We would like to acknowledge the financial support from AFC, from Saskatchewan Pulse Growers, and also from Pulse Canada. We are grateful to Loquat for providing the pea starch and to Cargill for providing the normal maize starch. This is a photo of our group taken in February. I think that's the end of my talk, and I would like to take any question in our Q&A session.
Great. Thank you, Yangfeng. It's really interesting to start to see some of those unique properties of faba bean coming out here. So I'm going to take some time quickly here to introduce our last speaker, which is Dr. Phyllis Shand, who is now a professor emeritus of food and meat science in the Saskatchewan, having worked there for over 30 years. She was raised on a small farm in Manitoba, and her undergraduate studies were at the University of Manitoba, followed by a Master's of Science from the University of Alberta and a PhD in meat science from Colorado State University. Dr. Shand's research interests are in the areas of meat protein functionality and gelation, meat quality and tenderness, and ingredient interactions in processed meat. However, she has been working on the functionality of pulse ingredients in meat products for almost 20 years. She's been a co-supervisor of 24 graduate students and currently supervises or co-supervises 2 MSc students and 1 PhD students, and has been active in outreach and extension to the food industry through participation in short courses, troubleshooting, and product development. She is the former chair of the Canadian Expert Committee on Meat and Meat Products and currently serves on the board of directors at the Saskatchewan Food Industry Development Center. So with that, I'll pass it along to our final speaker here. Thanks, Phyllis.
Hi, I'll start again since I had muted myself. Anyway, thanks very much for that introduction. Today, we'll be looking at our sensory profile development for Faba and other pulses. Now, certainly there's been much greater interest in using pulses and pulse-derived ingredients in food product development, but many consumers are not familiar with pulse flavor, and that may lower acceptability of new products. As Julie started out today, she said we must delight the consumer. And so we do want to put a smile on the face of consumers when they consume new pulse-based food products. And the picture on the side is that of my niece enjoying her blue Jell-O. Now, while we want to put a smile on the face of consumers of pulse-based ingredients, I don't think the blue color that was seen in this photo has anything to do with pulses. There have been some concerns about off flavors, and I do remember receiving a sample of Faba protein concentrate, which affected the flavor of some sausage we made. And this was something that we just did in the pilot plant about 15 years ago, and we really didn't have any knowledge of what could have caused that. So certainly looking at off flavors is important with respect to pulses, but maybe let's go back to the really beginning of what is the flavor profile of various pulses. And so in this current research, we wanted to develop a standardized sensory evaluation lexicon for pulses and pulse-derived ingredients. For those of you who haven't heard the term lexicon before, it's a set of descriptors with definitions and reference standards. And this is something that would be used by an experienced or trained panel to really get to the root profile of a food product. In this work, we did some descriptive sensory evaluation of various pulses, comparing them to Faba. And then we looked at Faba bean cultivars and derived products. This was limited to the cotyledons of these 3 Faba cultivars. So it was looking at the effect of removing the seed coat. Now, first, our work began with developing a sensory lexicon. And this grew larger part of the study, in part due to COVID, since we were unable to do in-person sensory work for over 2 years. So based on the literature, we looked at over 120 studies that had done sensory work on pulses, and we collated all of the terms or descriptors that were used in the various studies. And these were grouped into 4 categories. We looked at whether they looked at the basic tastes, such as bitter, sweet, salty, sour, and umami. We also looked at the aroma attributes that were measured by smelling, the flavor attributes as perceived in the mouth, and also something called trigeminal feeling factors. So the sensation in the mouth that you get not from an actual flavor, but from the physical contact with the muscles in your mouth. So astringency is an example of that, where you get that puckering feel when you come in contact with certain compounds such as tannins. So when we collated the terms, we found a great number that had been used, such as beanie, brothy, metallic, floury, sulfur-like, fruity, cheesy, roasted, greasy, fatty, or spicy. Bitter was the most used basic taste, identified in about 12% of the studies. Beanie was predominantly used to describe the aroma and flavor in 43% of the studies. When we look closer at what is beanie, based on the literature, it is a very complex term, yet it's poorly defined. And we found that there were a number of sub-descriptors that were falling under the term beanie. We did identify several knowledge gaps. Certainly there was inconsistency in the studies in the use of terms, and even in the presence, or whether or not people looked at aftertaste. We also compiled a list of food and chemical standards that were used as references. So this part of our work resulted in a publication in the journal Comprehensive Reviews in Food Science and Food Safety, and we developed a sensory wheel based on the frequency of use of various terms. And so some of the subdescriptors we found for beanie, for example, the half basically of the chart in orange, first they were not specified, it was just called beanie. Some of them referred to a naming of a pulse, so they would say lentil-like or chickpea-like. They also used terms such as green, earthy, nutty. And then at lower frequencies of use, terms such as musty, grassy, cooked bean, raw bean, potato, pea pod, mushroom-like, or brown. Basic tastes were used in some of the studies. Most frequent, as I've already mentioned, that of bitter, but sweet was mentioned, sour, salty, and umami. There were a few studies that looked at aftertastes, and the particular ones that stood out were bitter and beanie. And then a very small number of studies actually looked at attributes such as astringency and spiciness. So this was a sensory wheel that was collated based on the literature. And so in our later work, in our current work, we want to further refine this wheel for use by the industry. So our conclusions from this part is bitterness and beanie are very common descriptors. Many studies had developed their own lexicon, which made comparison among studies difficult because people were using different words in different studies. And so we see that there's a need for more standardization of terms for use by both the scientific community and industry. We also need increased rigor to get appropriate and reproducible sensory data. And the use of standards can be valuable in doing this. And it also would be useful to follow sensory properties through the value chain in a standardized way, rather than sort of the fragmented way that we see in some of the literature today. The next part of our work actually did involve getting down to tasting of pulses. And so for this work, we started with looking at the flavor profiles of whole seeds, and also of some seeds with the seed coat removed. And in order to really look, we wanted to look at a wider range of pulses. And so we selected 6 different pulses. I'll show the next slide with some more details. And in order to provide these in a cooked form to the descriptive panel, they underwent a pre-treatment, with soaking for 16 hours, steam blanching for 3 minutes, and then they were hot filled. They were then pressure cooked at 15 psi for 75 minutes and in jars. And after that, just before presentation to the sensory panel, the product was pureed and then was served to our sensory evaluation panel. We also did some other properties such as measuring particle size of the purees and some chemical analysis. But today I'll focus just on sensory evaluation. In order to sort of get a good range of descriptors, we selected 6 different pulses to get that are popular to compare, and 5 different ones that were compared to Faba bean. We also worked with the three Faba bean cultivars that you've heard about previously by other speakers. So for this work, we had to train our descriptive panel, and we wanted the panelists to be able to perceive and identify various aroma characteristics, basic taste, trigeminal and flavor attributes, and then to rate their intensities. And for this training, we used a build-up method where we introduced certain properties in the first training session, added more properties as the panel became comfortable and with those particular attributes until they were given the entire set of descriptors that we were using in this study. The training was done in an open classroom so that we could separate panelists well during this part. There was a discussion at the end of each training session. At the end of training, they were given a performance evaluation with a repeated set of samples evaluated. So for this work, we started with 19 panelists and we retained 16 to go on to the actual study. We did use various reference standards, and here are some examples of the reference standards that we found useful in this study. For grassy, we used freshly cut green grass. Luckily, this was done in the spring, so we could find some outside. We used green. For green, we had crushed thawed garden peas. For a cooked pulse-like attribute, we had pureed white kidney beans that were from a can. For floury starchy, we used raw white wheat flour. Metallic was the reference standard was a copper coin on the tongue. And for astringent, we used a 5% solution of liquid wine tannin. Some of these reference standards were strictly for aroma, while others they could taste as well. Now, we had poor success finding standards for some of the terms, and so we, for example, sulfury, and so actually we dropped that term in part because we couldn't find a good standard for it, and secondly because it was in very low frequency use based on the literature. Serving was done. Samples of the purees were standardized to a common total solids content. They were served warm in random order, one at a time, to the sensory panel. And the study was carried out in our sensory evaluation booths under red lighting to disguise any color differences among the pulse purees. So here we evaluated 15 aroma attributes and 16 taste, flavor, and trigeminal attributes for the panelists. The scale used was 0, not perceptible, to 10 being very intense. Now, I won't have time to show very much of the data results. There's a lot of data here, but I'll just point out a few features in the comparison of the 6 pulses that we tried. So the first attribute I'll mention is that of pulse-like. They all pulses evaluated had the highest value for pulse-like. For sour, one pulse stood out, and that was Faba bean. For green, the yellow chickpea and green line yellow pea stood out as having more green aroma notes, and the various beans stood out as having more earthy flavor aroma notes. Move to the other side of this slide, and here we have one more attribute on the top, which is that of astringency. And you can see this was a property that did have that was in higher intensity. The next bitterness also stood out, and here the black bean and the faba bean stood out as being higher in these attributes. Umami was an attribute that was relatively low from aroma, but umami was then much more prominent, especially for the chickpea and the pea in the cooked flavor of these products. Earthiness also still stood out for the various beans. Now, I have done some comparisons of aroma and flavor, and in the interest of time, I will just show these very briefly. So here is green lentil. The solid line is the flavor, the dotted line is aroma. And so you can see that aroma and flavor were of similar intensity, except umami taste was higher. And sweet also differed for chickpea, where it was higher by tasting than by aroma. For black bean, you're starting to see a little bit more bitterness showing up. For the yellow pea, we see that umami was greater in yellow pea than in black bean. And also the flowery note was higher. For the faba bean, we are seeing a bitterness showing up in flavor and some umami, and as I mentioned previously, greater earthy. The pinto bean was sort of intermediate in properties, but what was interesting was nutty was showing as more intense than some of the other pulses. Just show you one slide now of some of the attributes and the analysis that we did on the three different faba bean cultivars. So if we look at pulse-like flavor, for Malik, Snowbird, and Fabel, all three were relatively similar in pulse-like flavor, and there really was no difference between the flavor of the whole seed puree versus the flavor of the cotyledon puree, showing that there was very little effect or very little to no effect of seed coat on that flavor property. Bitter taste was quite interesting. Actually, Snowbird had the highest bitter taste, and that was whether or not we removed the seed coat or not. So there are bitter components in the cotyledon as well as contributed from the seed coat. Malik was intermediate, and the sample with the lowest bitter taste was that of Fabel cotyledons. Astringency was a very interesting term, and we actually found that for Malik, the highest astringency was found in whole seed of Malik, while for Snowbird or Fabel, they were slightly lower than the whole seed of Malik. So what's interesting is bitter taste somewhat followed that of astringency, but there were some subtle differences in that astringency was actually greater in samples such as Malik whole seed than the comparable bitter taste of that puree, indicating that we can't always say that these always go together. So our conclusions for this part were that the sensory wheel provided a good foundation for lexicon development. We also developed a set of useful reference standards. We now have mapped the aroma and flavor profiles of 6 popular pulses. Often aroma intensities predicted flavor by mouth, but not always. And we need to look and consider astringency more. It's a maybe underreported attribute. Now, we did see that Fabel cotyledon purees had the least bitterness, and Snowbird, with or without the seed coat, had the highest bitterness, which was unexpected as it is a low tannin variety. And so that needs further evaluation. So in summary, we now have a sensory lexicon and protocol to share for training of descriptive panel for evaluation of pulses and pulse-derived ingredients. It'd be useful to now move further through the value chain. We certainly do need to further analyze our sensory data and compare it to chemical analysis data. And a next step in the future might be to hold a sensory workshop to engage sensory panel leaders. So with that, I list my acknowledgements, and I certainly want to mention the collaborators on this work, Dr. Claire Chidwidir and Dr. Gina Wanasundra, and the funders of the work and the contributors who provided samples for our work. Thank you with that.
All right. Well, thank you. That is so much information to digest. So thanks to all of our speakers here this morning. We're going to take some time to go into some audience questions. I just wanted to go back to our co-moderator, Julie Mann here, just to get your perspective. So having listened to all of this research, do you mind giving your thoughts on how this relates back to the food industry?
Yeah, so I mean, all the speakers, I mean, amazing work you're doing, amazing job to bring forward kind of I would say the pieces of the puzzle that we're missing right now. I would say Faba is poised for growth. We're in a stage where we're a little bit less developed maybe than the pea industry. But I think when you look at varietals and like obviously varietals matter. I'm just going to give you a couple highlights because I know we want to get on to questions. The process driving the final product. So whether we're looking at flours or whole bean or concentrates or isolates, I think bioactives are a space that is not being highlighted in a lot of the other plant protein areas. And maybe we need to be doing a little bit more of that because I think that brings a very different nutritional perspective. Structure function to application, as we've heard today, critical understanding what you have, what you're going into, and what you're trying to produce. If you don't know that, you're probably going to struggle. And then I would say just, partners are needed, and don't think about Faba as the only ingredient. I think as I've been in this space for, 20 plus years, I think protein blends are always a good way to think about the industry. Each one brings its own benefits. And you really need to leverage putting them together to see what you can achieve from function, nutrition, complete nutrition, organoleptics. So I think if we think about that, we can also balance out the protein and some of the other issues that we have in different protein spaces. So I'm going to stop there, and I would love to hear what Q&A we have on the deck.
Great, thanks, Julie. So the first questions that we got are a little bit more related to the analytical techniques that were used as part of this research. So specifically, if you can provide some more insight into measurement of visine, convisine, as well as phytic acid for some of these samples.
I believe that question is for me, right? Yeah, visine and convisine in Saskatoon Research and Development Center, we have developed a method based on literature, and it is a HPLC, liquid chromatography based method, and phytate analysis that is based on megazyme phytate kits, which is again based on what we practiced over the years in lab based on literature, and that method is not now available as a kit from megazyme, so we use that, so that way it is comparable with some of the industry measurements.
Great, thank you.
It's a spectrophotometric method. Yes, FIDATE, and you can use a microplate reader, so can do more samples at a time.
Perfect, thank you. We have two questions here for Lamia, it looks like. So just one point of clarification for the antioxidant activity, if you were using whole faba bean seed or a fraction, as well as if you can comment on the ability to measure the anti-diabetic activity of some of these proteins, specifically if you're looking at things like measuring blood glucose response.
Okay, thank you for the questions. For the first one, in the particularly of our study that we have used the whole flour. So, and we simulate kind of after we eat the flour by doing the in vitro digestion. So it's the antioxidant activity was measured at the end of the digestion on the digestate, and we targeted the peptides in this situation. But to make the answer short, it's the whole flour that was studied in this study. Yeah.
Great. And do you have any comments perhaps on the glucose response measurement?
Okay. Yeah, for the anti-diabetic activity, actually we used we targeted the enzyme, the dipeptidyl peptidase 4, which has a role in the metabolism of the glucose. So there are also a lot of medication that target these peptides as an anti-diabetic medication. So the assay was based on measuring the capacity of the generated peptide on inhibiting this enzyme. So this enzyme actually degrades some of these incretin and indicator that have that reduces that increase the production of insulin and reduces the glucagon. So it has it improves the it controls better the glucose level in the blood. But we didn't do it by measuring in vivo any blood level sugar or something like that. Yeah.
Great, thank you. So a couple questions here specifically related to resistant starch content. So Yangfeng, do you expect similar results if you were to look at a dried powder that was extracted from the boiling water for some of these starches?
Yeah, thank you, Claire and Long, for your questions. I understand this is a very important topic, and I would like to, Mary, can you share the slide I just uploaded when I was listening to Phyllis talk? I would like to share the slide to explain why the processing is so important for the low glycemic of the pulses. Because if we really like look into the literature and when we hear a lot from like the research articles or even from the news, pulses, they are known to have a low glycemic benefits. Then definitely it's related to processing, but based on my understanding, I think the first thing I want to share with you is like in my presentation, if you really look at the starch per se, when we talk about isolated starch, based on our work, either from in vitro or in vivo, I don't believe the starch itself is the main reason. Instead, when we look at the seed structure, as you can see on this slide, we believe the seed structure, like when we compare pulses with a cereal called ledin, they are the main reason for the low glycemic benefits of the pulses. So I'm very grateful that Claire and Ron asked the question. So when we look at the pea and the faba bean, you can see, actually Dr. Janetta Wanasandra already mentioned this in her presentation. The starch granules in pea and the faba bean and other pulses, they are kind of encapsulated in a cell. You can see the starch granules, they are embedded in the protein and the fiber matrix. In contrast, when we look at the maize and barley on the right, actually they are called ledin, are packed with starch granule. They don't have the protein and the fiber matrix. So what does the protein and the fiber matrix do here? Based on our understanding is when we have this matrix structure, we do have the data to support it. So the key message here is when we have this kind of matrix, they function as a physical barrier to delay the digestion of the starch. So that's the reason for when we eat the whole bean or whole pea or the faba bean, most of the time they will show a low glycemic benefits. But definitely another reason is because of the relatively low starch content of the pulses. Because usually pulses we are talking about 40 to 55% starch, but for most of the cereal grain they may have 55 all the way to even 90, for example, in rice. So I hope this is the key concept I want to share with the audience after I read Claire and Ron's question. So to answer Claire's question is, if we extensively process it and then we destroy the cell wall structure, then we won't see the high resistance starch content of the pea and the faba bean. But if we only have the whole pulses and then powder them, and when we try to grind them, but not severely, I believe the resistant starch content will be retained. That's key information. But once we acidated the starch, as you already see from my slides, and also we have another study. So we ground our pulses like a pea, faba bean, lentil into fine flour. And then we have the rice as a control. When we use them to prepare the pet food, actually in beagle dogs, their glycemic response among all the different pulses and also rice, they are almost the same. We didn't see any difference because when we grind it and then when we extruded the sample, basically we destroy this kind of structure as you can see on the slide. So we lose the localized benefits of pulses. I hope that I convey the message. Then for the baked good, this is also a very good question because baking is all about how much moisture is there and also the baking temperature. So if we don't have enough water, if we don't have bake it to really high temperature, actually if we can retain the granular structure, the raw state of the starch, actually I believe the pulses they will have more resistant starch. So actually some of the data from Dr. Chang Liu at AFC in Guelph, they published some paper recently. They add some of the pulse flour. Most of them are like a dry bean powder into bread. Actually, they did show that the replacement of the wheat flour with the dry bean and other pulse flour, they show low glycemic benefits. That's in bread. I'm also thinking for the cookies, because cookies is a very interesting system. It's also baked good, but the moisture content is very low during baking. And also we have a very high level of the fat. So with that, previous research also showed that even we use a wheat flour for the baking, actually more than 80% of the starch was not gelatinized. So to answer Ron's question is, I believe if we have the like a really limited amount of water and we didn't bake it to very high temperature, I believe we can retain some of the raw porous starch granule if we use the porous flour for baking. And then I believe the resistant starch will be retained. And then we can also convey the low glycemic benefits to consumers. I try to just link things together. This is really related to processing and what's the level of the disruption of the protein and the fiber matrix, and also what's the level of the starch gelatinization.
Great. That was okay. We have another question for you here. Just if you can speak to any effects of enzymatic modification or other chemical modifications and how that might affect some of the properties of starch.
Yeah, sure. Actually, this is one of the main research focus of our group. As our presenters already share with us today, a lot of time when we try to extract the protein, then we have the starch as a byproduct. And I think Post Canada also have done a lot of work to promote the utilization of the different post starches. So in like short messages in our group, we try to find some of the unique ways to modify the different starches. We did try like one enzyme called mutagenic alpha amylase. So we were able to reduce the retrogradation. Also, we were able to double the resistant starch content of the faba bean and the pea starch after cooking. But that's still not enough for industrial application. They want more. Like we can increase the resistant starch content from about 6% to 12, 13%, but that's still too low. So we are still working on using some other modification. For example, we try to combine like the extrusion with enzyme to see if we can diversify the functional property and improve the resistant starch content. Chemical modification is still one of the main methods to modify starch, particularly for food and some other industrial application. But for the food application, because everyone want a clean label starch now, we didn't do too much. But I believe in theory, all the different developed chemical method for starch modification can be used for the purpose. But I think the last comment I want to share with you is we can definitely diversify the functional property easily for starch or protein. But another thing is really for the varialization of the starch. So we want to really make the market, make the ingredient to all the way to the end of the market. I think we need to think about the cost. Like enzyme, a lot of time very expensive. Like the enzyme may be even more expensive than your starch. So definitely in terms of like scientific work, a lot of interesting to explore. And we are trying to use more enzyme and the physical method to modify starch. And hopefully some of the ingredient we can see in the product development or even in some of the industry uses for the post starch, which is really a main byproduct from the post processing industry.
Thank you. So it looks like that is the end of our questions here. So I wanted to thank everybody for joining us here today, and a special thanks to all of our speakers here. It was very informative, a lot of information. So just a reminder that we will be circulating the recording to all registrants here after the event. But if you have any questions or if you want to get connected with some of these researchers here, please don't hesitate to reach out to me. My contact information is available on the Pulse Canada website, and we do have a lot of other resources there related to pulse utilization. So thank you everyone for coming, and we'll see you next time.